A is close to population B, it is benefit for population A to be here with population B, yes? What is minus or negative? It means when population A is with B, it is negative for population A. It is not beneficial. Population B has adverse effect on population A, so it is negative. 0 means no interaction, no difference. Okay? To be with this population, there is no difference. For example, here you can see one population B benefit to be with population A, but population A, no difference, no importance to be with population B. Okay? So these symbols we have, and we want to talk one by one about different interactions. First interaction between population A and B is called neutralism. It is called neutralism. What does it mean, neutralism? Population A, no effect to be with population B, and population B, no effect, no importance to be with population A. Now we want to have some examples about neutralism. When two populations are close together and there is no importance, there is not benefit, not positive, not negative. It is look like you're feeling. Sometimes you sit on a chair in the classroom, one person sit beside you. So if you are happy, it means it is benefit for you. It is not only for eating, it is according feeling. What is the feeling of population A about population B, and what is the feeling of population B regarding population A? Sometimes one person sits beside you in the class and you like to change your place. Why? Because you feel it is a negative interaction. You don't like to sit there. Sometimes one person sits beside you and there is no importance to be there or not to be there, not positive, not negative. Not positive, not negative. Now we go through first interaction as neutralism. Neutralism. What is that? Two microbial populations without any interactions. How it possible? How is it possible two different populations to be in neighborhood and there is no interaction? Sometimes it is this, between populations with extremely different metabolic capabilities. Suppose you have two children Put one population in one flask and the other population on the other flask, and put them close to each other. So there is a barrier between them. There is a barrier. How can they have any interaction? When there is barrier, there is no interaction. Or suppose in a habitat, in soil, one population in one place and another population on another place. They are far from each other. When they are far, their interactions tend to neutralism. But it is not 100%, 99% it is so. As much as they are far, for example, two sisters living in one home have more interactions, or two sisters in two different countries. As much as two persons going far from each other, their interactions minimize. But it is not 100%. Suppose there is a tree on the garden with a stem 5 meters up and 5 meters roots down. Some microbes are 5 meters down and some microbes 5 meters up. They are very far from each other. We think the most probable their interactions are neutralism. 99% is so. But suppose if the microbes in the soil attack to the root and the tree will die. So the microbes on the soil, the pathogenic plant pathogens, have interaction, negative interaction on the microbes living on the leaves on the tree. Okay? So as much as two populations to be far from each other, interactions going to zero or neutralism. But in some exceptions, maybe even though they are far, they can have some interactions. Physical separation alone doesn't ensure a relationship of neutralism. Example, pathogenic microorganism invade a plant root, resulting death of the plant. So, destruction of the habitat of other microbial populations on the leaves of the plant. Microbial populations, when they are outside their natural habitats. See, life is in the earth. Earth has three habitats. One habitat is soil. Second habitat is water. Water of oceans, of gulfs, water of lakes, water of rivers, and the third habitat is air. In soil, there are many native microbes. In different types of waters are many types of native. Air doesn't have any native microbes. So What are the microbes present in the air? All present microbes on the air come from soil and water. From wind, many soil particles go up to the air. From cough and sneezing of humans, of animals, from infected plants, many bacteria, many viruses, many spores come to the air. From wind, many aerosols, many droplets of water come to the air. So in air, we don't have any native organism. When two populations come into non-native habitat, their interactions tend to neutralize. Suppose a wind coming and two populations from soil come to the air. Their interactions, because this habitat is not a good place for their life, because their metabolism comes down close to zero. So accordingly, their interactions come to zero. Suppose you pour some soil to the river. So those microbes of soil cannot have the maximum metabolism in the water. So if they have any interaction in the soil, cannot have it in the water. Because when one microbe going from soil to water, going from water to soil, going from salty water to fresh water, going from fresh water to salty water, or going from soil or water to air, their metabolism comes to the closest to zero. As much as metabolism to be less and more less, so their interactions comes close to the neutralism. Neutralism between two microbial populations when they are outside their natural habitats. Example, in the atmosphere where population densities are low and all microorganisms can be considered alloctonous or non-indigenous. Their interactions tend to zero. Environmental conditions that don't permit microbial growth favor neutralism. River or a lake in the summer, there are many interactions among different populations present. But in the winter, if it is very cold and the lake or river is full of ice and water is freezed or frozen, what happened? When the temperature come down to zero or below the zero, we know the metabolism come down to zero. As much as metabolism come down, so the interactions tend to neutralism. Why do you use refrigerator at home? You put milk in the refrigerator. Why? Because you know when the temperature comes down close to 0, 1 degree, 2 degree, 3 degree centigrade, the metabolism of bacteria in the milk come down. And as much as their metabolism tend to 0, their interactions to another microbes, living things, and non-living things tend to their neutralism. So there is no interaction with another microbes, and there is no interaction with the milk. Sometimes you want longer reservation. So you put some food in the freezer below 0, sometimes minus 20 degrees centigrade. Or in some places, in special cases, they use minus 80 degrees centigrade. So the metabolism is very close to 0. So when there is no metabolism, there is no interaction. So you see, for a long time, you can have your food in the freezer without any growth of microbes. When we say neutralism, so neutralism is there is no interaction of microbe with another microbes and microbe with non-living environment, non-living substrates present in the food. Environmental conditions that don't permit active microbial growth favor neutralism. For example, microorganisms frozen in an ice matrix, such as in frozen food products, or microbes present in polar sea ice or frozen lakes. What happened? So don't permit active microbial growth, and it is favored to neutralism. Number 1, 2, 3, and right now it is the time to talk about number 4, resting stages of microorganisms. Suppose there are many predators attack to the prey. For example, suppose Paramecium attack and they start eating bacteria. Maybe for bacteria, food is okay, there is not toxic substance, but they are under the pressure of change their appearance from vegetative form to the spore form. So what happened? So predators like to eat vegetative form of bacteria. They don't like to eat spore. So when bacteria are vegetative form, what is the interaction? The interaction is predation. There are some predators engulf and eat prey. Now there are Paramecium and spore. What is the interaction? The interaction is neutralism, because spores have no interaction to Paramecium, and Paramecium or Paramcia don't have any interactions to a spore. And it is so for cyst of protozoa. Cyst. So number 4, resting stages of microorganisms. The low metabolic rates don't force them for competition. Spore cyst, etc., any resistant forms during environmental stress like heat, drought. In any case, when one microbe changes their appearance from vegetative form to what? To resistant form. In such a situation, its interaction with other microbes tend to neutralize them. Okay, first interactions over. Neutralism, population A 0, population B 0. Now we are going to the commensalism. One population 0, no difference, but the other population get positive interaction, benefit from the interaction. Benefit. What is that? Commensalism. Commensalism. Mensa is a Latin word. Mensa is a Latin word, means table or food table, and com means common. Common means a food table used for two populations. Now we want to see what's happening. Suppose sometimes you see there is a lion. Sometimes I give you some example from animals, from general ecology. Then I continue with microbial ecology because you can understand it very well. Suppose there is a lion going and find a prey like a deer, like a sheep, like what? And eat. When it's hungry finished, then lion goes. Don't stop there. There is no difference for lion. Nobody come to eat the remaining meat, or wolf coming, or birds coming. There is no difference. But it is very important for others to go and eat that remaining food. So, for example, you go to a restaurant, you eat food. Some of us, we take the remaining one home, but some, or in some situation, something remaining, and we go. And after us, after going, the waiter coming, and maybe eat the remaining food, or give it to the poor people, or give it to the animals outside. So it is no difference for you who eat the remaining food from you. So you are zero. It is not important for you. But for waiter, for poor people, for animal, for whatever, use the remaining food is beneficial. It is positive. Okay? Some microbes are present in our large intestine. We eat food. We digest in small intestine, but some not used material going to large intestine or sending them out as feces, as stool. If microbes use the remaining one, is no difference for us. If nothing used them and completely going out of the body, it is no difference for us. So there are some comments present in our large intestine. They use some not used food from human. Not obligatory. Okay, now some examples. Some microbial examples. Number one. One population modified the habitat in such a way that another population benefits. What this part wants to say. See, suppose you have a nutrient brush in your hand in a tube, and you inoculate an obligate anaerobe and then shake the tube. When you shake the tube, this medium is full of oxygen. So this anaerobic bacteria cannot grow. Okay? Agree? Now you inoculate a facultative anaerobe. So facultative anaerobe can grow in the presence and in the absence of oxygen. So now you have two populations. You have two populations. One population is obligate anaerobe, cannot grow because you shake the tube and the tube is full of oxygen. The other population is facultative anaerobe. It can grow in the absence, even though in the presence of the oxygen. Now what happens? Facultative anaerobe start using oxygen. As much as facultative anaerobe using oxygen, the situation will be better and better and better for obligate anaerobe because oxygen is reduced during the time. So for facultative anaerobe, presence of obligate anaerobe is indifference. If obligate anaerobe is not there, facultative using oxygen. If obligate anaerobe is there, again they use oxygen. So for facultative anaerobe it is no difference. But for obligate anaerobe, the presence of facultative is very important, because after some time, with absence of oxygen, they can grow and multiply. Okay? So for facultative anaerobe is 0, or it is not affected, but for obligate anaerobe is beneficial. One population modified the habitat in such a way that another population benefits, because the modified habitat is more suitable to its need. You can hear this interaction. We are talking only regarding to oxygen. We don't talk about food. We are talking only regarding to the need or no need of oxygen. Population A, facultative anaerobes, utilize oxygen and lowers oxygen tension. Then population B, a habitat suitable for growth of obligate anaerobe. Population A, unaffected. Population B, benefited. Commensalism continues as long as two populations don't compete for the same substrate. So, the presence of population E is beneficial for B, and A is not affected regarding to the B population. This interaction is commensal till the time food is sufficient and they don't compete with each other. Is it clear? Did you understand? Is it any question? Okay. Very good. Production of growth factors forms the basis for many commensal relationships. See, when you are working in the lab, sometimes you can prepare simple media for growth of bacteria, for example, nutrient broth or nutrient agar. But sometimes the bacteria which you are working with, they need enriched media like blood agar, chocolate agar, serum agar. These media are enriched. Why? Because some bacteria can produce organic substances, many necessary things from simple material, many structural parts from simple things, all amino acids from single things. But some bacteria are defective or production of some necessary materials from simple material. For example, one bacterium cannot produce 2 or 3 amino acids. So if you give it in the medium, it can grow. If you don't give it, it won't grow. We call them fastidious bacteria. Fastidious bacteria. Sometimes at home you can see, for example, whatever you prepare at home, your child says, I like it, I like it, I like it, yes. But sometimes one of your child says, I don't like it, I don't like this, I don't like that. So you have many problems with fastidious children. In some bacteria, because they don't have necessary enzymes, they don't have necessary genes. Because of the problem in genes and enzymes production, they don't have capability to produce some necessary, some essential things. They are facultative. In the lab, You give them. Who give them in the environment? Who give them in the water? Who give them in the soil? It is the question. Production of growth factors forms the basis for many commensal relationships. Production of growth factors. Most microbes produce growth factors and some extra excrete outside themselves. For example, population 1 excrete growth factor, vitamins, amino acids, excrete to the environment. Population 2 utilize this growth factor and can grow. If population 2 is present or is absent, population A do this. So it is indifference. It is neutral to be with population 2. But population 2 is happy to be with population 1 because without this cannot grow. Population 2 is dependent on vitamins and amino acids which produced and excreted by this group. Example, Flowobacterium brevis excretes cysteine and Legionella pneumophila can use it in aquatic habitat. If Legionella is present or is absent, Flowobacterium excretes cysteine. So presence or absence of Legionella is neutral for Flowobacterium. But Legionella is very happy to be with Flowobacterium because they need cysteine. Okay, so Flowobacterium is in this interaction, Flowobacterium is 0 and Legionella is plus or positive interaction. Note, production of growth factors and excretion allow fastidious microbial populations for growth and survival. So in the lab, we give them whatever they need, but in the environment, in the water, in the soil, production of growth factor and excretion by many microbes allow fastidious microbial populations, growth and survival. Another example. Transformation of insoluble compounds to soluble ones. So you know bacteria don't have mouth. They receive soluble food by diffusion, by active transport. But mostly foods are inaccessible. They are insoluble. So some bacteria can change insoluble compounds, insoluble foods to soluble ones. Conversion of soluble compounds to another forms also. Changes of the state from solid to liquid and liquid to gas. Example. Population 1 in sediment produce methane. In any case, this population 1 do this. But population 2 need methane. Population 2 needs methane. So population 1 is 0 in this interaction because if population 2 is present, or population 2 is absent. Population 1 do it. In any case, population 1 do this interaction, do this job. So it is 0 in the interaction. But population 2 is happy to receive methane and use it. So population 2 plus population 1, 0. Yes, and this interaction is commensalism. Another example. One bacterium with the name of Desulfovibrio can change sulfate and lactate to acetate and hydrogen. If methanobacterium is present or is absent, Desulfovibrio do it. So Desulfovibrio in interaction with methanobacterium is 0. That methanobacterium is very happy to be with Desulfovibrio to receive acetate and hydrogen in combination with carbon dioxide can produce methane. So Desulfovibrio is not happy, not sad. No difference. Do its job. But methanobacterium is very happy to be with the Desulfovibrio. It is another example of commensalism. Another one, population make a compound available to another population without actually transforming the particle compound, mostly in soil. It is the same previous example as, for example, in the soil there are many phosphate. Phosphate is very essential for growth of all the cells. Phosphate is necessary for human cells, for fungi cells, for protozoa cells, for bacterial cells, for all cells. Phosphate is very important. Why? Suppose one bacterium want to divide or one human cell want to divide. So chromosome should be double. Chromosome constitutions are nitrogenous bases, yes, and the oxyribose, sugar, and phosphate. How can one bacterium divide without replication of DNA? So if all the things are present in a medium, but there is no phosphate, so there is no growth. Why? Because without replication of DNA, it is impossible of multiplication of the cell. Another thing, one cell wants to be double. So cytoplasmic membrane should be synthesized. What is cytoplasmic membrane constitution? It is phospholipid. How can human cell or bacterial cell or fungi cell to be divided going to mitosis or binary fission without biosynthesis of phospholipid? Suppose one cell has 1000 ATP, for example. Now this cell wants to be double. How it can be double without double in the ATP, adenosine triphosphate? So phosphate in enzymes to produce small molecules like glucose, sucrose, maltose, lipids, and then ingested to blood and receiving to the cells. Okay? So mostly microbes, pathogens, and non-pathogens, they have food in front of them, but these foods are very big. They should produce exoenzymes to change it to the small size molecules or small size substrate by their exoenzymes, extracellularity. So population 1, conversion of organic molecules to substrates. If population 2 is present or if population 2 is absent, population 1 do it. So population 1 is 0 in this interaction, but population 2 is happy because population 1 used the substrate and the remaining substrate is used by population 2. So population 1, no difference, unaffected, and 0 interaction. But population 2 is happy and positive interaction. For example, population 1, like most fungi, by extracellular enzymes, convert polymeric compounds like cellulose into simpler compounds like glucose. So if population 2 is present or absent, population 1 changing cellulose to glucose and use it with many extra remaining glucose. But population 2 is very happy of the presence population 1 because population 2 lacking cellulase. But now when it is in neighborhood of fungi can utilize those complexes because cellulose changed to glucose and they don't have problem with glucose. Another example, removal or neutralization of toxic materials. Removal or neutralization of toxic materials. Suppose population 1 doing oxidation of H2S. H2S is very toxic for population 2 and H2S can kill population 2, but it is used as a food or something like that for population 1. If population 2 is present or is absent, population 1 do it. So population 1 in interaction is neutralism or 0. But population 2 is very happy of presence of population 1 because by detoxification, population 2 can continue its life. Population 2 is H2S sensitive anaerobic microbial population. Population 2 is a microbial population which is sensitive to the presence of H2S. Example 2, precipitation of heavy metals. So when heavy metals are present in the water, it can kill many microbes. So one population can precipitate it. So make the water clean. So it is detoxification. it can be done by populations such as sulfate reducers. If the other populations are present or absent, this population do precipitation of heavy metals. So their interactions are zero. They are unaffected. But the other populations during the toxifications are very happy to be with this population. Or production of volatile mercury compounds in aquatic